Well, welcome everyone. I'm Jen Farmer from the Friedreich Ataxia Research Alliance, and my colleague Kellen Madden and I are excited to host today's webinar with Lexeo Therapeutics. This is our second webinar in our FA Awareness Month series, so this is great. This will be an opportunity to get an update on the Friedreich's ataxia cardiomyopathy program. This is a gene therapy pipeline program, and we've got two guest speakers today. We have Greg Aubert from Lexeo Therapeutics, and we have Dr. Z from the University of South Florida. So we're going to turn it over to Greg and Dr. Z, and Kellen and I will be here to help moderate the Q&A. If you have questions as we go along, as information's being presented, feel free to put it in the Q&A box down at the bottom of your screen. Try not to use the chat feature, try to use the Q&A feature. And when we get to the end of the presentation, we'll open it up for question and answer. And with that, Greg, I believe I'm turning it over to you, right?
Thanks, Jen. Hi, everyone. It's a real pleasure to be here today to give this update. So as Jen told you, I'm Greg Aubert. I'm leading the clinical development for this Friedreich's ataxia cardiomyopathy program. I still practice cardiology at Loyola University in Chicago, where I see cardiovascular genetics patients. And with me today, Dr. Zajiewicz from the University of South Florida will present some of the data we're going to share with you today. So let me start with some disclosure here that you can read through. It's a bit what I just say. I'm an employee of Lexeo. But then if we go into the topic, the today presentation, right? First, we'll have an overview why and how the heart is affected in the Friedreich's ataxia. Then we'll discuss a bit about gene therapy approach and the early finding from the Friedreich's ataxia cardiomyopathy clinical trials. There is two ongoing with this gene therapy, and I will tell you more about that. And then there will be the next step, the Clarity FA Natural History Study. We tell you that we'll start enrolling very soon this month or the next one. And finally, some question and answer. So if we start with this overview, right, I'm sure most of you know the frataxin gene is involved in making energy for your body, right? So the frataxin gene tells your body to make an important protein that is called the frataxin, which works in the mitochondria. So the mitochondria, you know, they're the small organelle within the cell. That's really where all the energy is made, and then help the cells having the energy to sustain its function. Right. So with Friedreich's ataxia, there's a change, a mutation, there's some repeats in this frataxin gene, and that at the end lead to a decrease in frataxin protein level within this mitochondria. So the mitochondria doesn't work properly. And so the body cannot make enough energy, and that's obviously this energy is important for every tissue, but specifically for some, like the brain, the muscle, and the heart, where they need a lot of energy to sustain their function. So why does the heart matter in Friedreich's ataxia? You know, the heart is the leading cause of death in people with Friedreich's ataxia. And without Friedreich's frataxin protein, there is a lot of things that cannot happen. As I told you, the heart is a machine that works 24/7 and really need to have a lot of energy. And so all this energy is coming from the mitochondria. And so if the mitochondria do not work properly, and the heart has to work harder to make up for it. So that's added stress on the heart. And what we can find is there is a thickening of the heart that is called hypertrophy, or a heavy heart, right? And this thick heart can cause some problem. One can be that it's stiffer, so you can have issue to relax. You can also have some finding where then you're gonna have some issue to contract. But all those things taken together are called cardiomyopathy. And up to 80% of people with Friedreich's ataxia gonna develop this cardiomyopathy. So that's why it's really important as part of the team, and I'm sure you know, to having a cardiologist that is there as a care team, because he can monitor those findings when a cardiomyopathy develops as part of this Friedreich's ataxia. So then how we could treat that, right? And that's where we're gonna discuss this gene therapy approach. So there are currently no approved treatment for Friedreich ataxia cardiomyopathy specifically. So the usual treatment for other type of cardiomyopathy, hypertrophy, you might have heard, probably would not help at all for Friedreich ataxia because they really target different things than the mitochondria, where is the problem in Friedreich ataxia. So one approach to treat the heart there in Friedreich ataxia is the gene therapy. So what is gene therapy? So the gene therapy is a way to treat, or even sometimes to prevent, the impact of a genetic disease or genetic mutation by either fixing or replacing a gene that is not working properly. In this case, it's really replacing, because we know in Friedreich ataxia, there is not enough frataxin that is made. So the goal will be really to try to go bring another copy of the gene to re-express the gene in the cardiomyocyte. And so the gene is frataxin. And we're gonna insert a functional version of the gene in the cells. And then that's where we need a vector, right? So you have the cargo that's really gonna take this gene and bring it to the heart, to a specific target cell. And you may have heard there's different kind of virus, most often the AAV virus, that you have different letter after 8, 9. In our case, it's RH10. And in our hand, this RH10 is very good to target the cardiomyocyte. So we use a vector that we know will go to the heart and then express the gene in those cells. So if we go a little bit more in more detail about this virus, so as it says here, it's a AAV-RH10H FXN, so human frataxin. So it's an investigational gene therapy designed to deliver a functional frataxin gene to the heart cells. So as you can see here, that's the whole capsid, so the virus with the gene inside, is given intravenously into the circulation. You're going to target primarily the heart, but can go in other organ. And then once in the heart, then the gene will go into the nucleus and express a functional frataxin gene that will play the role of frataxin within the mitochondria and help with that. So now we're going to go to the early finding we have from the phase 1/2 trial, and I'm going to soon give the mic to Dr. Zdziewicz, but just wanted to let you know the data you're going to see is an aggregate of 2 studies that are going on at the same time. So today there's 17 participants that had been dosed with different doses of this AAV-RH10 on one time. So on the left, you see there's the very Cornell investigator initiative trial that is sponsored by the NHLBI, so the NIH, and it is Dr. Ron Priestad that run this study. Is also the founder of Lexeo. And then they have 2 doses, a low dose and a mid dose for now, and they're still enrolling in this study. And then aggregated with that is the Lexeo sponsor trial with the same gene therapy with 3 different doses, low dose, mid dose, and the highest dose here. And that is completed now. And now I'm going to give it to Dr. Zdziewicz for the data.
Okay, well, thank you. So as Dr. Aubert said, there are 2 different clinical trials running, but they're very similar trial designs, and they allow us to look at the data together. So for these trials, we enrolled people from 18 to 50 years of age who needed to be diagnosed with Friedreich's ataxia with some evidence of cardiomyopathy. And all of the trial participants received the one-time intravenous infusion of AAV-RH10. And what Lexeo is looking at is, number 1, safety always. The amount of the frataxin protein in the heart, only in the Sunrise FA study, not in the Cornell study, because that necessitates a cardiac biopsy. Other measures of heart health, and of course, measures of function like neurologic function and other reported outcomes. And the study length is 52 weeks or 1 year, and patients are followed up for 4 years. Next. Okay, so, all 8 patients in the Sunrise study have been enrolled, and they've been through the study. No participants have dropped out of the study, which is great. There were no major signs of immune reactions in the blood, which can occur with any gene therapy program. Last fall and summer, there was 1 participant in the Sunrise trial that developed myocarditis, which is a heart inflammation that was found on MRI 1 year after the gene therapy dosing. the patient did not have any symptoms. This came about because of routine labs that were taken and further investigation. The cause of the inflammation was not identified. Of course, 1 year after, it's possibly related to study treatment, but again, she was asymptomatic for any problems. And she's doing well. She continues to be monitored, and she's responding well to current treatment. Next slide. Okay, so in the Sunrise FA trial with 8 participants, all participants received a heart biopsy that showed increases in the frataxin protein levels in the heart. So in the graph you can see here, these are the frataxin levels in the heart 3 months following treatment. So we did heart biopsy before treatment at baseline, and then 3 months after treatment to check for frataxin protein levels. All of the 8 participants had increased frataxin in the heart after gene therapy treatment, as you see here. The low dose patient, 1 participant, had a 29% increase. The mid dose patient that we had 3 participants. They had a 69% increase. And the high dose patients, which were 4 patients, had a 115% increase in frataxin, as evidenced by the cardiac biopsies. Next slide. And also there are improvements in measures of heart health. And when we talk about heart health, we look at the left ventricular mass index, LVMI, and troponin. So as Dr. Aubert said, the left ventricular mass index, LVMI, measures the heaviness of the heart. And the heavier your heart is, the more it's associated with a worse outcome in Friedreich's ataxia. Because so many patients get cardiomyopathy with Friedreich's ataxia. So in this case, the lower the number, the better for the patient. And most patients did show an improvement following gene therapy in the heaviness of the heart, in the LVMI mass index, particularly those who had heavier hearts before treatment. So you can see that there were 6 participants who had heavier hearts at baseline, and then they had a 27% improvement in the LVMI, the heaviness of the heart. But in all 12 participants, there was a 15% improvement in the left ventricular mass index, which is very important. And then on the other side of the graph, you see the average troponin I improvement in the trials to date. So 11 participants. Troponin is a marker of heart injury. We measure it in the blood test. And of course, in this case, the lower the number, the better for the patient, because troponin indicates high troponin indicates heart injury. All participants showed improvement after gene therapy, which was very impressive, 68% improvement, except the 1 individual who had the myocarditis and a heart inflammation at the patient's last visit. So you could see that it went from an average of 537 to an average of 173, which is a 68% improvement in troponin I. Okay. There were also improvements in measures of function, neurological function, and reported symptoms. So this is the FARS rating scale. This is the clinical rating scale that we use for Friedreich's ataxia patients. This is what we call a modified clinical rating scale, or the FARS, as well as as a cardiomyopathy questionnaire. So let's look at the FARS. The average mFARS improvement to date in the patients was 2.75 points. So the modified Friedreich's ataxia rating scale is a tool that neurologists and ataxia specialists use to measure how Friedreich's ataxia is progressing and how it's affecting movement and coordination over time. It ranges from about 0 to about 100, 120. The lower score reflects improvement. A change in 1 point is considered clinically meaningful because FA patients usually progress about 1 to 2 points a year. So there was an improvement of 2.75 points in the 12 participants. In terms of the Kansas City Cardiomyopathy Questionnaire, or the KCCQ, this is a survey that helps doctors understand how someone's heart condition affects, their activities of daily living or their quality of life. In this case, a higher score is improvement, and a change of 5 points is usually considered a clinically meaningful improvement. So for 5 participants in the Sunrise FA trial only, there was a mean average of 6 points in the KCCQ after the study. Okay. I want to thank Lexeo Pharmaceuticals, FARA, my entire USF Ataxia research team, Dr. Arti Patel, Debbie Hoffman, and Fornaz Tabatabian. These are cardiologists who specialize in FA and immunology and heart transplant, this is not, but they've done a lot of work, and it's been a really exciting time and just amazing work. So thank you very much.
Thanks a lot. Before we go to the next slide, I also wanted to share with you that Weill Cornell is still enrolling in the study and is funded by the NHLBI, so the NIH. And then if you have any question about Cornell trial, we wanted to reach out to them either by email or by phone. Now we can discuss what comes next. Next step for LX 2006 investigation gene therapy program. So there is some exciting and good news, right? This Lexeo plan to start a new study, probably early next year, end of this year, to support regulatory approval from the Food and Drug Administration. So for the study that we plan at the end of the year and early next year, participants will be given the investigational gene therapy, LX 2006, the one we saw the preliminary data. And FDA has agreed with our plan to include both children and adolescents in this study. And finally, the FDA has also agreed that we will not be required to have a placebo arm in this study. And that's why we're going to discuss about this Clarity FA, which is a natural history and can replace a placebo control arm. So data collected from this separate natural history will help support our submission to the FDA. So we will start first with this Clarity FA as soon as this month, and then later this year, early next year, we will start another trial where patients will be actively getting the investigational drug. So what is a natural history and why it's important? So in order to develop treatment for a disease, we researchers in the general medical community must first learn about it. So natural history is a type of research that tracks how the disease developed and change over time. And we observe symptoms, changes, pattern to really better learn what to expect from the disease, find a way to diagnose it earlier, and how to manage the disease better. And information from natural history studies provide important knowledge that can help develop better on new treatment for the disease. So it's important to note too that patients that are in the natural history are not getting any investigational study drug. And on top of knowing all those things about the disease for this specific case here, it's really to have natural history that we can compare to when we have our study where we're gonna give the investigational drug. So what about Clarity FA, this natural history? So the goal of this Lexeo sponsor Clarity FA natural history study is to learn how the heart develop and worsen in individuals with FA, and ultimately to be used to control and to understand how good is the treatment in the one that will receive it in the next study. So it's really important to know that, because even though approximately 80% of people with FA will develop heart muscle disease, we still don't know exactly how those things progress. And there is some research that have been done. It is always good to have more. And for our case, to really look at the exact same things that we're gonna look in the trial later on. So information that are gained from participation in this study will definitely provide valuable data to learn more about heart disease in FA and help advance our gene therapy. And additionally, data collected from Clarity FA will be used to compare to the gene therapy study. So who is eligible? So it's exactly the same kind of people will be eligible to the natural history, then later to the treated study. So it's open to individuals who need a genetically confirmed Friedreich ataxia diagnosis and an evidence of cardiomyopathy. So as we discussed earlier, a thick heart already. Clarity FA will include 2 age groups that really gonna match the age group we're gonna have later in the treatment study. It's individual who are at least 16 years and old, so the 16 and the 17 will be in the adult part, and then individuals that are younger, between 6 and 16 in another cohort. The full eligibility criteria will evaluate by the study doctor to determine if individual is the right candidate for this study. It's a global study. There will be multiple locations across the United States, but later there will be also a location around the world. And then for you to know, patients that are taking Skyclarys are allowed to enter this trial, or they will be allowed to enter the other trial too, but they need to be at least for 12 weeks prior to starting the study on the stable dose of Skyclarys to make sure there is no change that we can observe that are due to starting the Skyclarys. So what to expect for this natural history? So first, there's a screening period to make sure the patient is matching the criteria and can enter. And then there is 4 visits. Baseline, visit 2, visit 3 that can be done remotely, and the visit 4. And what we measure here will be the same exact measurement that we do later for the study with the investigational drug. So it's some lab tests, some imaging, some questionnaire, and some safety assessment. So the imaging is mainly done with cardiac MRI to really measure precisely the size of the heart and how it evolves over this year. So the study doctor will learn about your heart disease through those information and performing those routine study assessment. And again, for clarity, there won't be any gene therapy received. So then more information, as always, for any clinical trial, participation in Clarity FA study is voluntary. You may withdraw from the study at any time for any reason. If you refuse to participate or decide to withdraw, you will not suffer any penalty, loss of right, or loss of benefits to which you are entitled. There will be no direct payment for taking part in the study, but the participant and their caregiver will be reimbursed for reasonable study-related expense for travel, right? If you need to go to the site, stay there, hotel, and so on. As with any clinical study or trial, there may be risk involved with participation, and then we will have to sign a consent that will explain those risks. We encourage any individual considering participation in a clinical study to consult with your physician. And then, so if you want to know more, there will be in the QA, obviously, after, but if you're interested, if you know which site will be open, when, and so on, please reach out to us at clinicaltrial@lexeo.com, and we can orient you for where either answer your question or where those trials happen. And finally, wanted really to emphasize that for us, connecting with the Friedreich Ataxia community is always a chance, right? At Lexeo, we really try to do everything what we do. So it's really at the heart for the individual with Friedreich Ataxia, but also their loved one, and really proud to be partner for many years now with FARA to support those impacted by FA. And then together we can potentially push the boundaries of the science and achieve shared goals to address unmet treatment needs in Friedreich Ataxia cardiomyopathy. Thank you all for your attention, and we're gonna be open for your questions.